Greetings, my name is Yanda Gumede and I'm presenting on behalf of Group 11. The neurotransmitter that I'm presenting today is domic acid. Okay, first things first, what is domic acid? Domic acid is a natural occurring toxin which can be produced by certain type of algae. Domic acid can be harmful and even lead to death in humans if contaminated shellfish is consumed. The toxin of domic acid was detected in the year 1991 on the west coast. Domic acid is tolerant in change in temperature and many other acids, meaning cooking or freezing shellfish does not destroy domic acid in the shellfish. It can be fatal to people if consumed in high doses, since there is no antidote for domic acid, which causes condition called amnesia shellfish poisoning. Symptoms of domic acid within 24 hours of ingestion are as follows: vomiting, nausea, diarrhea, and abdominal cramps. As a result, neurotransmission is being affected in the way it functions since domic acid shares structural similarities with glutamate, an excretory neurotransmitter in the central nervous system. Domic acid binds to and activates the carnic acid glutamate receptor, allowing sodium influx and negligible amount of potassium outflow, leading to neuronal depolarization. These are some of the symptoms of domic acid. On our left, we have a person who wants to vomit. This can be an amnesia since he is not vomiting. Then on our right, the other person in the picture is in the toilet and showing signs of having stomach cramps as well. These are also signs of shellfish. The following symptoms take place because of neuronal depolarization: headache, dizziness, confusion, loss of short-term memory, motor weakness, seizures, profuse respiratory secretions, and cardiac arrhythmia. Glutamate. Domic acid is a water-soluble amino acid that causes toxicity by stimulating the glutamate. So what exactly is glutamate? Glutamate is one of neurotransmitters in the brain. It is the most abundant neurotransmitter. Glutamate can bind to 4 different receptors. This allows it to have a major presence and the ability to stimulate and communicate with other nerve cells. Glutamate is involved in more than 90% of excitatory functions in the brain. Functions of glutamate include hearing and memory, energy source of brain cells, chemical messenger, sleeps, wakes, eyelash manager, pain signaler. There are several routes by which domic acid has an impact, but glutamate receptors are the one that should raise the most alarm. Glutamate is a neurotransmitter in the brain that activates glutamate receptors. Domic acid is an excitatory amino acid counterpart of glutamate. Domic acid binds to these receptors with a high affinity, which causes excitotoxicity by acting iteratively on inotropic glutamate receptors on both sides of the synapses and blocking the channel from fast desensitization. Moreover, endogenous glutamate and N-methyl D-aspartate receptor agonists work together synergistically to cause excitotoxicity. Domic acid specifically harms the hippocampus and amygdaloid nucleus in the brain by turning on AMPA and kainate receptors and resulting in an influx of calcium. It harms the neurons. Although the entry of calcium into cells is natural, an unchecked rise in calcium leads to cell deterioration. Short-term memory loss happens because of possible serious hippocampal damage. A recent study in mice found that even at levels deemed acceptable for human intake, it may still harm the kidneys. 100 times lower concentration than what the FDA permits causes kidney damage.
Domoic acid is a biotoxin that's produced by an algae called Pseudonychia, and these harmful algal blooms, as we call them, occur with warm water temperatures. It enters the ecosystem and is consumed by smaller fish. So when sea lions eat these fish that have been eating this algae with this toxin in it, it builds up in their systems, and the toxin binds to receptors in their brain and causes seizure-like activity. And one of the most frustrating parts of my jobs is seeing these animals suffer from this horribly debilitating disease that has no real treatment. But what we can do is help provide supportive therapy for the brain to prevent that permanent brain damage. The condition of domoic acid intoxication in sea lions is similar to a brain disease in humans called temporal lobe epilepsy. What we learned from California sea lions and their conditions and what happens in their brains can be applied to this human condition, and vice versa.
The important part of glutamate. Glutamate is also needed for making another neurotransmitter in the brain called gamma aminobutyric acid, GABA. GABA is a calming neurotransmitter which is involved in sleep, relaxation, anxiety, regulation of muscles functioning. Glutamate is stored in the wall vesicle called synaptic vesicles, located at the axon terminal, which is at the end of each nerve cell. As a signal travels along a nerve cell, the electrical off signal causes the vesicle of neurotransmitters, in this case glutamate, to be released into a fluid-filled space that is between the nerve cells. This space is called synapse. On the other side of the synapse is the next nerve cell. Glutamate must bind to specific message receiving receptors. Toxic effects of shellfish and neuron. It is proven that human exposure to domic acid surfaces through the intake of contaminated shellfish that has accumulated the toxin while filter feeding on toxigenic phytoplankton during blooms. Its toxic effects are behavioral abnormalities, seizures, and neuron degeneration limbic system. Mechanism of DOMA activates the subtype of receptors for the amino acids of glutamate, which leads to oxidative stress-mediated neuron cell death. Domic acid acts as glutamate antagonistic and is excitotoxic in the vertebrate nervous system and other receptor-rich agents. Together, experimental research and medical investigations have demonstrated that DA exposure largely affects the hippocampus regions of the brain, is linked to seizures, and impairs cognitive function. A distinctive feature of neurobehavioral signature of DA is the presence of both temporary and long-term memory function alterations that resemble human anterograde amnesia. Adult non-human primates used in experimental research have shown that DA is a dose-dependent emetic that causes clinical and neuropathological alterations associated with excitotoxicity, memory loss, and behavioral hyperreactivity during mid-dose therapy reflect changes in arousal and/or emotionality. Higher levels of the A therapy cause blended neurotoxicity, which is evident in the form of convulsions, status epilepticus, and animal mortality. The cycloserine. This was originally introduced as an antibiotic to treat tuberculosis, has also been observed to possess anti-anxiety properties. It is a partial NMDA organized as the glycine modulatory site which can facilitate the opening of calcium channels attached to NMDA receptors and appears to have a role in the activity underlying land association instincts of fear. Its properties are that it acts as an antibiotic and anxiolytic, also has a huge role in neural activity. It may accelerate the therapeutic effects of cognitive behavioral therapy in the OCD patient, and the greatest treatment effect may be limited to uneducated cases. So obviously very important that response may not be as good if the patient is on traditional medicine used to treat OCD. Nerve cells and communication signal continues its way from one nerve to another. So, how does D-cycloserine works? It is believed that D-cycloserine has an impact on specific receptors found in the amygdala region of the brain, particularly those known as NMDA receptors, which are connected to fear. The phobia is not directly addressed by it. Rather than that, the medication seems to encourage the parts of the brain which manages the process of eliminating fear reactions. To clarify, it has the effect of accelerating or enhancing the response of exposure therapy. As a result, it can reduce the typical inpatient experience in the clear stages of treatment and prevent individuals from quitting therapy permanently. Clinical trials have shown limited adverse effects in the use of D-cycloserine for treating anxiety disorders, though further research is necessary. Additionally, its efficacy has been studied in the treatment of conditions such as Alzheimer's disease, schizophrenia, OCD, PTSD, and other anxiety disorders. So this is the end of our PowerPoint presentation.